In this topic, we're going to discuss the control of the cardiac cycle. So, by the end of this topic, you should be able to answer the questions: how is the cardiac cycle controlled? So we can look at the sinoatrial node, the atrioventricular node, Purkyne tissue. Then we're also going to look at the pressure and volume changes in the heart. And we're going to end off with the electrocardiogram. Now, for the heart to function efficiently, there must be careful control of the sequence of events that takes place during the cardiac cycle. So within the wall of the right atrium, so look at this diagram, and the right atrium there's a distinct group of cells called the sinoatrial node. So in this diagram, it's labeled SA node. It's a specialized patch of tissue made of myofibrils and nerve endings. So it's in the right atrium. It's myogenic, which means that the stimulus comes from within the muscle itself. It has a basic rhythm of stimulation that determines the beat of the heart. So it's for this reason it's often called the pacemaker. A wave of action potentials is initiated in the cells around the sinoatrial node, and it spreads through the network of fibers across both atria, as you can see in this diagram here. This causes the atria to contract. Now, a layer of fibrous connective tissue separates the atria and the ventricles. This is called the atrioventricular septum. It prevents the excitation wave crossing into the ventricles. There's a gap in the atrioventricular septum that comprises a group of cells that can conduct the excitation wave. This is called the atrioventricular node, AVN. After a short delay of about 0.1 to 0.2 seconds, it conveys the wave of excitation to the ventricles along a series of specialized fibers called Purkyne tissue. The Purkyne tissue is organized into a specialized structure called the bundle of His. This is where the action potential is transmitted along the base of the ventricles, where the bundle branches into smaller fibers called Purkyne tissue. So the wave of excitation is released from the Purkyne tissue, causing the ventricles to contract quickly and at the same time, from the apex of the heart, which is the bottom of the heart there, upwards. Now, cardiac muscle cannot respond to a second stimulus until it's first relaxed. This is called the refractory period. Something to note is that the cardiac muscle will not become fatigued or go into a state of tetanus. So here's a lovely diagram to show you the wave of excitation as it passes from the sinoatrial node to the atrioventricular node, down the Purkyne fibers in the interventricular septum to the apex of the heart. This will cause the ventricles to contract. Right, let's just run through what we've just discussed. Describe how the cardiac cycle is controlled. So the signal is received in the sinoatrial node, which is the pacemaker. Then what happens? Action potentials spread through the atria. The atrioventricular septum prevents the signal going to the ventricles, except through the atrioventricular node, AVN. Excitation moves through the Purkyne fibers in the bundle of His, and this initiates the contraction from the base of the ventricles upwards. Right, let's discuss pressure and volume changes of the heart. Blood pressure varies between a maximum, called the systolic blood pressure, when the ventricles contract, and a minimum, called the diastolic blood pressure, when the heart is relaxed. So we're going to look at the pressure and volume changes that occur in the heart during a typical cardiac cycle. So systolic blood pressure is 120 millimeters Hg normally, and diastolic blood pressure is usually about 80 millimeters Hg. So two important points to remember before we look at the graph. Pressure and volume in a closed container are inversely related. So an increase in pressure results in a decrease in the volume. And blood moves from a region of high pressure to a region of low pressure. So here you've got a graph showing the pressure changes in the different vessels over a period of time. Take a moment to have a look at the different curves and the shape for the aorta, the ventricle, and the atrium. So in an exam, how will you remember which curve is which? Well, think of where the pressure will be the highest. So you can see that peak, the orange curve is for the ventricle. The ventricle will have the highest pressure, so their peak will be the highest. There'll also be very high pressure in the aorta. And the atria, not so high. So let's identify where systole is occurring in the heart. This is where you're going to look for the peaks. So looking at the orange line, where do you think ventricular systole is occurring? As you guessed it, those two peaks there, that's ventricular systole. What about atrial systole? So this occurs just before ventricular systole, so it's the little bump before the big bump. So now that we've identified ventricular and atrial systole, we need to identify what's happening in the atrioventricular valves. The atrioventricular valves are found between the atria and the ventricles. So take a moment to think about during which events they will be closed and when they will be open. So we're going to be looking at where the ventricular and atrial graphs intersect. So where that orange and green line intersect. So the first intersection is where the ventricular pressure is increasing as the ventricle gets ready to push the blood out into the arteries. Here you don't want the backflow of blood, so the atrioventricular valves will be closed. As the pressure decreases in the ventricle and it goes below the atrial pressure, the atrioventricular valves open. Now let's have a look at the semilunar valves. Where do you find these? These are found between the atria and the arteries. So in this graph, the aortic pressure is the graph all the way at the top, that blue line. So where the ventricular pressure and the aortic pressure intersect, something should be happening at the valves. So when the ventricular pressure increases as it pushes the blood into the aorta, the semilunar valves will be forced open. As the pressure in the ventricle decreases on the other side there, the semilunar valves will close. So now that you know the key features, let's discuss what's happening in each graph, starting with the ventricular pressure, so that orange line. Ventricular pressure is low at first, but then it gradually increases as the ventricles fill with blood because the atria are starting to contract. The left atrioventricular valve closes and the pressure rises dramatically as the thick muscular walls of the ventricle contract. As the pressure rises above the aorta, blood is forced into the aorta past the semilunar valves. So the semilunar valves are open. The pressure falls as the ventricles empty and the walls relax. Okay, let's have a look at the pressure in the atrium. So atrial pressure is always relatively low because the thin walls of the atrium cannot create much force. So it's highest when they are contracting. It drops when the left atrioventricular valve closes and then its walls relax. The atria then fills with blood, and this leads to a gradual build-up of pressure until a slight drop where the left atrioventricular valve opens and some blood moves into the ventricle. Okay, moving on to aortic pressure. Aortic pressure rises when the ventricles contract as the blood is forced into the aorta. So the semilunar valves will be open then. It then gradually falls, but never falls below 12 kilopascals. So this is important to remember. This is because the elasticity of its wall, the aortic wall, which creates a recoil action, is essential if blood is to be constantly delivered to the tissues. The recoil produces a temporary rise in the pressure at the start of the relaxation phase. Okay, here's a detailed graph of the different pressures and volume changes that occur in the heart. So take a moment to identify each stage in the graph or each vessel. So the aortic pressure is at the top. The ventricular pressure is the one with that huge peak. And then the atrial pressure is just beneath this. Now if you have a look, this graph also includes the aortic blood flow and the volume in the ventricles. So let's have a look at the ventricular volume. The ventricular volume rises as the atria contract and the ventricles are starting to fill with blood. Then there's a sudden drop as the blood is forced out of the ventricles into the aorta when the semilunar valves are open. The volume increases again as the ventricles fill up with blood again. Okay, lastly, let's have a look at the electrocardiogram. So during the cardiac cycle, the heart undergoes a series of electrical changes related to the waves of excitation, and this is created by the sinoatrial node. So the heart responds to these electrical changes. So if picked up by a cathode ray oscilloscope, these changes can produce a trace known as an electrocardiogram. So here you can see the different types of ECG during different heart conditions. Now, the different peaks in the ECG are represented by the different phases in the cardiac cycle. For example, the P wave is caused by atrial systole. Ventricular systole is represented by the QRS wave, and the start of ventricular diastole is represented by the T wave. So let me go through that one more time. The P wave is caused by atrial systole. Ventricular systole is represented by the QRS wave, and the T wave is the start of ventricular diastole. And that concludes our lesson. The end.